Yeah, thank you, Steve, and to Arwen for the invite to speak today. It is a bit strange talking to yourself in a room to a screen, so at least I've got Steve to look at, but I can't see many other people. But feel free to interrupt if you have questions as we go along. I don't mind that. So as Steve said, sort of my mission is really to identify new therapeutic targets in blood cancer, and in particular acute myeloid leukemia. And I do that by making use of several different omic platforms. And given that October is generally a sort of new intake for postgraduate research students, and this audience is quite broad, I think, in terms of career paths and stages, I've got quite a broad introduction, which will last about maybe 20 minutes. But then the latter half of that presentation will focus on a particular abnormality in AML. It's called RUNX1-ETO. It used to be called AML1-ETO. And I'm going to use that as the example of how we've identified new targets and how we progress towards translating them in the clinic. So Steve gave a nice introduction, and now I feel quite bad that I've got him in the stocks in this picture. But it was one of the few pictures we have of a group, and we were in Ash when we took this. And I have to thank really current members and past members, because really without their dedication to their research and their research degrees, perhaps lots of the targets we would have found might still be sort of undiscoverable at the moment. And I also have to thank Richard. I came here in 2000 to do a postdoc on RUNX1-ETO, and it was really through his guidance and tutelage and co-leading a program, sort of program of research funded by Blood Cancer UK, that we've managed to progress quite a lot of research, I think, in trying to treat what you'll see is a dismal disease, really, in terms of patient outcome. So currently we're about 10 to 12 researchers, and that includes Steve. He's instrumental in providing the clinical input into our projects. So as I said, I'm going to provide a background to normal hematopoiesis. So this is blood cell production. And then this sort of translational modeling approach is really, you can consider it a summary. I apologize to some of the group leaders here. It's a sort of more of teaching your grandparents to suck eggs type thing, but this is how we undertake our research, and hopefully it will have a bit of a hematology slant to it. And then I'm going to talk about RUNX1-ETO and finish off with mass spectrometry and how we identified something called CEB beta. Okay, so on to hematopoiesis. So I think this is quite a generalized audience, but if you weren't really aware, the hematopoietic system has served as a paradigm model really for how complex tissues are organized and maintained by this sort of somatic population of stem cells. So we call them the hematopoietic stem cell, or HSC, and you'll appreciate that blood is one of those highly regenerative tissues that produce these terminally differentiated cell types down here. Of course, we're interested in these monocytes and sort of myeloid arm of differentiation. So these cells self-renew. They can give rise to themselves and differentiate in what we sort of term a sort of bifurcation hierarchical model. But really, this concept is now being eroded a little bit by what we call this continuum of hematopoiesis. And what I mean by that is that this HSC pool now seems to have a lot of evidence through single cell sequencing and next generation sequencing that they're already lineage primed to be determined into sort of these terminally differentiated cell types down here. And a lot of this work began by someone called Irving Weissman in 1988, and subsequent studies in mice, where they tried to look at the molecular programming and the phenotypes of these early progenitor cells. And this has led to a lot of confusion in the literature and sort of non-standard use. So for the hematologists in the audience, more recently, Grant Channon, Eric Piatras, and the International Society of Experimental Hematology are now proposing that we sort of keep this continuum of hematopoiesis, but are restructuring the naming or nomenclature of these multipotent progenitor cells. But for me, and this talk, we're going to stick with this hierarchical process. And I can elaborate on that a little bit further into something that everyone's probably more commonly used to seeing. So this HSC gives rise to your terminally differentiated cell types down the bottom. And in what happens in leukemias, we get an accumulation of malignant white cells in the bone marrow and blood. And we can classify that in its simplest form into whether those cells that are leukemic are lymphoid in origin, or as my research goes, myeloid in origin. And what happens is that you can have something that's more acute. So this is aggressive. I think it would be rapidly fatal if left untreated, but it's potentially curable, and I'll talk about that shortly. Whilst chronic leukemias, on the other hand, they're a little bit more indolent, they're a little bit more slowly growing, but I think Steve would agree they're paradoxically a little bit more difficult to cure, probably due to that slow growth. So in AML, well, in all leukemias, there's some sort of genetic damage, but in AML, what happens is we get an accumulation of these leukemia blasts, and that's because we get this failure to differentiate. So AML is really considered a disease in block of differentiation, and I think you can see that more so here on some of these bone marrow slides that you can get. If you look around or look at some reviews, you get some good pictures. But this shows you an AML marrow. I think the characteristics here, for someone not familiar, is that they're clonal, they all look the same, they have the same sort of immature phenotype. Whereas if you compare that to the normal bone marrow, you've got lots of different cell types here representing different stages of development. So this would be a neutrophil, and this one here is something like a proerythroblast. It will eventually eject its nuclei and become one of these red cells. And this is what we want to see. We want to see the heterogeneity in these different cell types down here at different stages of development before they leave the bone marrow and enter the circulation. So my research has sort of been fortunate because if we can find the right tissue source, and for us that's neonatal cord blood, we can isolate these stem cells by the virtue of the restricted expression of this protein called CD34. And if we can isolate these stem cells, then perhaps we can grow these cells within the culture dish and examine how normal hematopoiesis occurs. And then we can modify that process when we look at abnormalities in AML. But why is AML important? Well, there's about 3,000 cases in the UK per year, and the incidence rises with age. And the peak incidence is around 85, I believe, and a median at 65. But importantly, if you look down to this Kaplan-Meier curve on the right, what you see is these sort of overall survival for patients with AML is dismal, and it's particularly dismal in those that are sort of over the age of 60 or less fit compared to some of these younger patients. And even in the younger patients, less than 50 years old, the cure rate is still, less than half. So there's a lot more to be done here, improving the lives for patients with blood cancer, and particularly the elderly who are often unable to tolerate current treatments. So another way of describing AML or classifying them is according to cytogenetics and molecular abnormalities, and that's important for clinicians. Steve can elaborate further than me, I'm a scientist, but what you end up using is this molecular classification to inform risk stratification, and that then informs the best treatment options. I just want to pay a nod to the French American British classification, or FAB, and I think most clinicians will agree now it's time to consign this to the history books. But as scientists, we still use it a little bit because whilst it's a subjective process and perhaps it's a dying art of trying to identify what is the correct FAB subtype by morphology, what we end up getting is something which resembles more of an undifferentiated AML in FAB M0 or M1. And this might be more akin to the hematopoietic stem cell. The comparator might be the early progenitor cell. Whereas if it's an M4 or M5, it resembles something that's more blocked in differentiation around the monocytic stage of development. And so maybe we should compare it to the transcriptional program that's closest to a normal human monocyte. So we do make use of this, and you might see that throughout the presentation. But as I've just alluded to, it's really the transcriptional the mutational landscape that's important in AML. And so far, over 100 molecular abnormalities have been identified. And I don't expect you to read this slide and try to identify many of them. But what I do want to point out is that I'm particularly interested in this one here. It's called RUNX1, RUNX1T1, but it's either RUNX1E2 or AML1E2. And it arises due to a chromosomal translocation a transcription factor called RUNX1. So how do we model this in normal human primary cells, and how do we translate our research? And so this could act as a summary to the targets that we've been identifying. But as I've said, I'm very much interested in RUNX1-ETO, and Richard has worked for a long time on another abnormality in AML called mutant RAS. And once we've identified our abnormality, the question becomes, well, how does that affect normal blood cell production? So one of the best models to use here would be a human primary cell, and we can isolate those, as I've already said, based on the expression of CD34. It's restricted to those stem cells. And if that tissue, like cord blood, has a relatively good percentage of cells, well, we're still talking less than 1% here, then we can isolate several thousand cells to transduce. So we use retrovirus, and these retroviruses are based on plasmids or vectors that will host your abnormality, say RUNX1-ETO, but will also co-express a selectable marker like green fluorescent protein. And equally, once we transduce these cells, we'd also be transducing in parallel an empty control vector shown here with green fluorescent protein. And we've been doing that now for about 20 years or so. And what that means is we're able to transduce greater than 70% of these cells, which means you don't really need to sort them unless there's a particular scientific reason to do that. And in addition, because we're using blood cells, these are single cells, so we can use flow cytometry and measure them on a sort of single cell basis. So the first step we want to do is try to determine the phenotype of the effects of RUNX1-ETO on blood cell development. And one of the best models to assay differentiation is this human primary cell model. Of course, we can look at other functional endpoints like self-renewal, migration, but we can use flow cytometry to determine particular stages of development within particular lineages within the blood cell. So what we found when we transduced cells of RUNX1-ETO was that there was a block in granulocytic development, and I'll show you that data later. But the next step was to try to determine the mechanistic understanding of that process. And traditionally, you know, around 2005, 2007, I used Affymetrix microarrays to do that. And we're trying to assay for changes in the transcriptome, such as, you know, mRNA, for example. But more recently, and, you know, coming down in price and ease of access, we're now using next generation sequencing RNA-seq, or even single cell RNA-seq in partnership with Oxford. But you'll soon see that mRNA doesn't always mean a change in protein. So we work with Manchester University, and in particular, Professor Tony Wetton, who encourages us to use something called SWAP mass spectrometry. And here we're looking for changes in the proteome. Either way, what we want to do is to try to identify key effectors of that phenotype, if you like. And we've identified several, and here are some of the current ones we're working on. CD200, for example, as you'll see later, is an immunosuppressive molecule expressed on AML cells, whereas CEBP-beta is more of a transcription factor known to regulate certain genes involved in differentiation and growth. So having identified these, what we next would like to do is to see if we can knock them down in our model system. So if they're overexpressed, we would knock them down and see what the consequences are in our model systems. Perhaps, as in the case of CEBP-beta, it's repressed by RUNX1-ETO. So some of the studies we would like to do is to re-express that gene of interest and see if we can revert the original phenotype. So all this is our sort of fundamental basic medical research, if you like, and what we really want to do is to translate those findings. So if there are potential clinical agents available, we will make use of them within our model system, but ideally what we want to do is to make use of them using primary AML material. And Cardiff has been one of the lead centres for collecting these samples. The AML bank is now under the purview of the Cardiff University Biobank, but the samples that come are high quality, they're viable, they have characterization in terms of clinical data and also next generation sequencing data and mutational analysis. So there's a lot of power of analysis that can be done here. And if you have a good project and the right funding, you can apply via CUB to get access to those samples and treat your cells with your clinical agent. And what we're hoping to do is to see if, at least ex vivo, we can provide some preclinical evidence that this is a target worthy of taking forward to early and late phase clinical trials. And I have one slide later that shows some of the data that we accrued for overexpression of CD200 in AML. So why do we need targeted therapy? Well, I think, as you've seen from one of the earlier slides, AML is highly heterogeneous. It's got all these molecular abnormalities. And to some extent, I think this limits the effectiveness of conventional chemotherapy. And on top of that, given that the disease is a disease of the elderly, not all patients are fit for high intensity chemotherapy regimens. So we need a better way of treating patients, something less harmful and less toxic. And to some extent, that paradigm's previously been proven. I was fortunate to be at ASH at the time when tyrosine kinase inhibitors were talked about, and the tremendous sort of changes in overall survival they had when they treated patients with CML. And there has been similar effects for AML. I mean, APL is a particular form of acute myeloid leukemia. It's called acute promyelocytic leukemia. And you can treat these patients with ATRA. It's also done in combination with arsenic trioxide. And this targets this particular translocation. And again, before ATRA and after ATRA, we sort of had this big improvement in several clinical parameters. Now I'm sort of entering Steve's wheelhouse a little bit here. So, I have to acknowledge him for the slide, and this perhaps is even outdated, but just prior to the pandemic, you could argue that the therapeutic landscape in AML was revitalized with the approval of a number of agents targeting different molecular abnormalities. And I think these went on to get approved by NICE. So there might even be more to this list since then. I'm not up with speed with the developments there, but I just thought I'd point out venetoclax because that's quite well known, at least in hematology, and it's a BCL2 inhibitor. And when you add that in combination with sort of non-intensive therapy, there were significant improvements. So I think the paradigm for looking for further targets is well established now, and that's where most of my work has come from for the last 20 years. And as I've said previously, we've used different omic platforms. And some of the more recent platforms we looked at was the metabolome, where we identified some key glycolytic mediators or key lipid mediators that are important in possibly, supporting proliferation of AML cells. So the ones in red are something we're actively working on at the moment through various funders. And so you might hear about these in various cancer themed talks, or even the Division of Cancer and Genetics talks, etc., or PGR Day. So for the last sort of 30 minutes, I want to talk about RUNX1-ETO. This is sort of a passion of mine. This is what I came to work on when I was a postdoc. So as that name suggests, it sort of has one element called RUNX1, and RUNX1 belongs to a family of transcription factors, and RUNX1 is frequently disrupted in acute leukemia. And I think that underlines its crucial role in maintaining the sort of integrity of hematopoietic development. You do see some mutations leading to other types of leukemia, such as TAL-RUNX1 in BALLL, but I'm really interested in this RUNX1 abnormality. And in all fairness, some of these abnormalities are quite rare. It's really the 821 that's the more frequent abnormality, and is often associated with better prognosis in AML. So, as the name suggests, it involves a translocation of AML1 or RUNX1 from chromosome 21 with the ETO gene on chromosome 8. And this results in the RUNX1-ETO fusion gene. There's been a suggestion now from Hugo that we have a double semicolon for translocations. If you weren't aware, that's something relatively new. Just makes it awkward to say. And we see this in about 12% of AML cases. And what's interesting was that patients with an 821 translocation, nearly 90% of them would be defined as a FAB-M2. So this really defined or implied the dominant effect of that translocated product on the emergence of that disease. So this is the translocation, And what you end up getting is a smaller proportion of RUNX1 is retained, but nearly the entire ETO portion is fused to RUNX1. But interestingly, if we look at normal RUNX1 and what its role is, it binds to this consensus binding DNA site here, and then it heterodimerizes with something called core binding factor beta. And this recruits then histone acetylases, which acetylates your chromatin and opens up and allows target gene expression of genes relevant to hematopoiesis. But when AML1 is fused to ETO, we don't really get the recruitment of these transcription factors, but what we do get is the binding of histone deacetylase or a co-repressor complex. And it's this co-repressor complex that keeps this chromatin tightly wound, if you like, and therefore you don't get transcription of the relevant target genes that are important in blood cell production. So when I started with Richard around 2000, what was not really known at that particular point in time was how RUNX1 might perturb normal hematopoietic development. So that was sort of my first job, if you'd like. It was to take those CD34 cells, transduce them with RUNX1 and then put them into different cytokine conditions. And this is a time-lapse video sort of trying to show that in a nice little video. So what we got is different cultures. This happens over 14 days. We have different cytokines promoting different lineages and development. And this is one of the easiest ones to see. I think what you'll notice is that this colony begins to pick up a reddish hue, and therefore it's becoming more of an erythroid colony. And this process is happening over 14 days, but of course, time-lapse speeds that up a little bit. But given that we can use more sophisticated techniques and flow cytometry, we can really unpick this on a sort of day-by-day basis if we really wanted to. So if I just get rid of that video. Okay, so back to our scheme, just to refresh your memory, we isolate CD34 cells, transduce them with RUNX1 or a control vector expressing GFP alone. And for the hematologists, we culture them in IL-3, stem cell factor, GM-CSF, and this promotes myeloid development. And then we can analyze the number of endpoints, as I previously discussed, by flow cytometry. So the first thing we wanted to do was to verify that we can indeed generate RUNX1 or AML1 progenitors. So this is our control, and this is RUNX1. What you have on the y-axis is flow cytometric data showing the expression level of CD34. And in each instance, you can see that nearly 95% of these cultures are CD34 positive. And we can infect over 70% of them with GFP. And if in this particular case, we would flow sort them to get a pure homogeneous population, and then we perform RT-PCR. And in our control, you can see we've got absence of AML1, but in our test, you can clearly see the presence of that translocated product. And Kasumi is a cell line acting as a positive control. So, that's what I did about 20 years ago now, but more recently, through the big efforts of Alexa Azevedo, she's a PhD student just finishing up and submitting her thesis. She performed some heroic CD34 cell isolations and isolated nuclear proteins from these samples. And it's heroic because in order to isolate nuclear proteins, you need several million cells. And to get several million CD34 cells is no mean feat. But she was able to do that and then Western blot, fractionate these samples into cytosol or nuclear and compare the RUNX1 transduced cells with control. And you can clearly see that we've got overexpression of RUNX1 at the protein level. So initially, we wanted to determine the effects of this oncogene on myelopoiesis, and we did that by first assessing myeloid colony forming capacity. So this is the number of colonies per 2,000 cells on the y-axis. We transduced our cells, plated them on day 3, and then analyzed them 7 to 14 days later. And the red is our control, and blue is AML1. I think what you can see here is that there's a 45% reduction in the ability of AML1 to form granulocytic colonies, but there was no effect on monocytes. And in fact, if we plated the cells a little bit later on day 6, we had a similar trend. But if you plate the cells later, what you're really doing is you're sort of losing that self-renewal potential. So what you've got in the red is the control cells are now not able to form these colonies, but in contrast, Runx-Juanito cells can, so they have an increased colony formation at this point. And this suggests that they're retaining some primitive phenotype or perhaps have self-renewal. And self-renewal is, perhaps for the new students that are starting, something that's characteristic, not of stem cells, but hallmarks of leukemia at least, and other cancers. So we wanted to next determine the basis for this failure in colony formation, and to do that, we examined the growth of myeloid cells in bulk liquid culture. And what this graph shows you now is the control cells, which proliferate and grow for about 3 to 4 weeks, at which point they become growth arrested and terminally differentiated. And when we look at AML Juanito, we actually get a completely different pattern of cell growth. You can't really see it here, but what you have is a 50% reduction in proliferation over the initial phase. And we now know that's due to changes in cyclin D1. But AML Juanito overcomes that initial cell cycle block and then eventually continues to grow at a rate similar to controls. But as opposed to coming to their natural end, these cells continue to grow for weeks and weeks and weeks, resulting in a 30 sort of fold expansion over our control cells. They don't go on forever, so they're not really fully transformed. They do begin to slowly stop. But I think what you can appreciate is that maybe these cells won't actually look like normal blood cells. And indeed, if we do morphology on day 17 on some of these GFP sorted cells, you can clearly see that Runx-Juanito cells do not look like normal blood cells. And what I'm comparing here is I'm really looking at these types of cells. These are granulocytes. You can see they've got this multi-lobed nuclear phenotype with some sort of hyperfine bridging in between those nucleus compartments. And this is completely absent in AML Juanito. And if you want to put some numbers onto that, what we end up observing is an 85% reduction in the ability of Runx-Juanito to make terminally differentiated cells. And this really leads to an accumulation of sort of intermediate promyelocytes. But we want to provide more evidence than just morphology. Morphology is subjective, even though it's quite clear in that particular instance. But we can use flow cytometry to analyze the differentiation status of cells as they undergo this process from HSCs to granulocytes. And over time, what we measure is CD13, which is a marker of myeloid development, and CD36. And when we combine these in a bivariate plot, we can get these sort of 3 populations. So we're interested in the granulocytes and the really bright CD13 positive, CD36 positive monocytes. Now, you have to remember that we're growing these cells under myeloid conditions. So eventually quite rapidly, what you'll get is a loss of this population because we don't include the cytokine erythropoietin. But you can modify the cytokine recipes and we can really dissect the erythroid cells quite nicely. I'm not going to talk about that today. I'm just going to focus on the myeloid aspect of it. So when we look at changes in cell phenotype, what we initially observe is that these stem cells rapidly lose CD34. So they're losing their early stem cell marker. And as they enter into, say, granulocytic development, they upregulate CD11B or will upregulate CD14 if it's a monocyte. So what did we get when we followed AML Juanito differentiation? So you can clearly see in the blue here that there was a higher proportion of CD34 positive cells. They didn't stay high, but they sort of delayed the downregulation of CD34. And this suggests that they're sort of early mature cells. But more convincingly, I would say, is that when you look for upregulation of CD11B, that marker of granulocytic development, it was significantly reduced. But again, you could say delayed. Eventually, these cells did upregulate CD11B, but never to the full levels that you'd see in the control cells. And we observed no difference in CD14. And indeed, when we looked at monocytic differentiation, within that compartment, we still observed changes in CD34 at the primitive stage of things. When we look for upregulation of CD11B, or in particular CD14 for monocytes, there was no difference. So to summarize, that's sort of, I would say, 3 years of work that we can summarize into this one slide here that resulted in these 2 publications. Effectively, RUNX1E2 expression is anti-proliferative at the very early stage of development, and it's conceivable that under those harsh conditions of clonal growth, it might result in a block in proliferation and hence a reduction in the colony forming efficiency. But when you plate those cells later, they do tend to have some level of self-renewal. Our assays are sort of limiting dilution single cells. It's not the gold standard. We would need to look at that in sort of transplantation in vivo assays. And I think the picture says it all really for that AML1E2 results in this blocking differentiation. So thus, our model recapitulates 821 human disease. It's a myelocytic leukemia associated with dysplastic granulopoiesis. And I think that model provides an ideal context for further study. So I think this was around 2007 now, we were interested in trying to determine what gene targets or what mRNA is switched on or switched off when you overexpress RUNX1E2. And at the time, little was known of those target genes. I think we now know a bit more about the genes, but we certainly don't know which proteins are affected by RUNX1E2. And that's the last part of this presentation. So we're interested to determine which ones are critical in maintaining normal development. So the plan was to follow the same model system I've described. Take your CD34 cells, transduce them with control and RUNX1E2, and then basically profile them immediately for changes in gene transcription. And we did this with an N of 4 for each condition. But given the power of our human hematopoietic primary model, we continued the growth of those cells until day 6, when we generated this mixed lineage culture. And through a series of lineage depletion using Miltenyi Max, we removed monocytes and erythroid cells and left a population enriched for granulocytes. And again, we can then put those into our gene profiling system. So what did we do with the data? So this was a while ago when different algorithms existed, and one of those was called MAS5. You can identify genes that are switched on or off by what they call present, absent, and marginal calls. And these are effectively quality control steps. So what we really did was we just removed genes that weren't expressed. And then we performed some threshold analysis. So what was the changes between control and RUNX1E2? So we arbitrarily picked 1.5-fold difference, because the next steps was to have some sort of statistical analysis to that, and even a non-statistical pairing that took into account the individual pairs of cord blood. So remember, this is done on an N of 4, so we've got quite a lot of power here. And what we wanted to argue was that we did these 3 different tests, and these are the gene lists that come up. But if the gene was truly changed, it would be changed in at least 2 of these different approaches. So the intersections here all form the 465 dysregulated probe sets, or genes, that we observed between RUNX1E2 and control cells immediately following transduction. And we performed a similar analysis then on our differentiated cells. So that would be the granulocytes, erythroid cells, and monocytes. Whereas I've already showed you that granulocytes is our sort of lineage of interest. So we looked for what genes were changed on day 3 and remained sustained changed on day 6. And when we did that, we identified 22 genes. And this was sort of our candidate gene list to take forward. So trying to provide some real life patient data into this data set, and not just via our model system, we wanted to look at these 22 genes and see if we can find some patient relevance. And this is where the online public repository data comes in handy, although at the time we had our own data. So we performed Affymetrix data already existed for 821 patients, and we compared them to non-821 patients and identified approximately 4,000 genes that were changed. And of the 22 genes we identified, 9 remained common with this particular list. And for us, that was an important list to take forward to see if we can validate them in subsequent assays. So to cut a long story short, these are the 22 genes. I don't think you need to read them all, but I will highlight some features of this table. 9 of them were identified with patients. And of those sort of 9, IL-17 and IL-10 receptor seem to be exclusively expressed in 821. And we thought that was it. This was going to be really important. We'll go back to our culture system. We'll add in some interleukin 17 or IL-10 and see what differences in phenotype we observed. But when we did that, we really didn't see any changes, which is a little bit disappointing. We were rather hoping that there would be a functional change. So we moved on to something different. We didn't look at a LOX5AP, but it was on our list. What's interesting about this is that it was downregulated across all different AML types. So this is an abnormality that could be applicable not just to 821 disease, but to other cell types. And I believe over the last 2 or 3 years, there's been some research into this gene, and it's involved in leukotriene biosynthesis. And some of our more recent data using metabolomics shows there's a massive change in lipid synthesis. So maybe this is something we'll come back to. But the genes we did follow up on were CD200 and gamma-catenin. So gamma-catenin is a homologue of beta-catenin, and Rhys Morgan was here, and now he's established his own group in Sussex. He's working on Wnt signaling in gamma. We're looking at binding partners of beta-catenin, and Adam Leckenby is a PhD student with Richard and myself, and he's coming to the end, and he's been looking at knocking out beta-catenin mainly, but along with gamma-catenin using CRISPR studies, and he's got some interesting data, so you might see 1 or 2 of his talks if you're interested in Wnt signaling. But for the majority of the time, we followed up on this molecule here, CD200, and I'm grateful to Steve Mann for helping us submit a grant and get that funded from what is now Blood Cancer UK, and Steve Coles and Steve Napper, who helped provide, well, Steve Coles was the postdoc on this project, and we generated a number of preclinical studies with the support from Steve, showing that CD200 is really important in immune suppression and regulation of immune suppression in AML. And more recently, Namrata Rastogi, she published last year some translational studies where we had a clinical agent blocking the effects of CD200. So just to touch on CD200 a little bit, what we see is this is increased in 40% of AML patients, not just 821. And if you have higher levels of CD200 shown here in the dotted line, then you're going to do worse overall in terms of overall survival for patients. And that's because the CD200 high AML patients have a reduced capacity to mount an immune response. And what we wanted to do was to see if we can block that immune response. So the AML cells, they have CD200, your immune cells, they have CD200 receptor. And if we can block that using a monoclonal antibody, for example, maybe we can revert the immune suppression. And Namrata took one of these agents from a company called Trillium Therapeutics, CD200.7, and took some AML blasts and co-cultured these cells with immune T cells. And we were able to revert to some extent, not all the way to the top, but revert the immune suppression when we looked at NK cell degranulation or interferon gamma assays. So we got to the point with CD200 where we're looking to put that into a clinical trial. I'm not sure if that will happen now, as some of the licenses and some of the industry or pharma focus has changed for Alexion and Trillium into other molecules. So then finally, for the last 5-10 minutes, I think I've got 5 minutes left, I wanted to talk about Runx-Juanito and moving our studies into proteomics. So as I said, RNA doesn't always equal protein. And since 2007, a lot of different studies have been performed using either single cell sequencing for more RNA targets, or they've been performed on cell lines, but no one's really looked at protein expression. And that's, I think, important because mRNA doesn't always correlate with protein expression, and mRNA alone is not enough to identify possible targets. You can find more if you look for changes in the protein. So we wanted to combine our previous data with some mass spectrometry data. So to do that, we work with Tony Wetton in Manchester, and he advised that with the advances in speed and mass accuracy, particularly with some of these latest triple time-of-flight mass spectrometers, that we can perform something called a data-independent acquisition. And this is termed SWAP, or sequential windowed acquisition of the total high resolution mass spectra. And I'm no mass spectra expert, but I think effectively what you're doing is, in old money, you would look for small windows, fragment this ion, and then go identify your fragment. But what you can do now, you can have, there's no need to rely on the detection of this precursor ion. You can detect them all in sort of one bigger window, if you'd like. And that would give you a variety of spectra. And it's the sort of advances in understanding this spectra and deconvolution where you can extract your now targeted data. So what that means is it's targeted proteomics, but it's quicker. And if it's quicker, you can use more samples and it can cost much less. So this approach was really more like 14 days, whereas this was more 2 to 3 days. And what we found was more advantage was that it creates a digital archive. And because it does absolute and accurate quantitation, you can quantitate your proteins across different mass spectra runs, and you can always come back to that data at a later stage and compare across different experiments if you'd like. So I think we were sold this as the sort of new Western blotting, if you'd like. So again, Alexa, who deserves credit for this study, and she's about to create a publication out of this, followed the same experimental design, transduce your cells with control and Runx-Bonito, 4 replicates each, and then isolate the proteins on day 3. The minor modification here was to fractionate our proteins into cytoplasmic proteins and nuclear proteins, because some of our previous data suggests that maybe protein localization is also an important abnormality in AML. Either way, save you the long data analysis, but we did threshold analysis and p-values, and we ended up identifying 183 proteins changed in the cytosol between control and Runx-Bonito, and 73 proteins that had changed in the nuclear compartment. And together, this identified 256 differentially expressed proteins. So we like to use enrichment analysis and pathway analysis, and I know a lot of people use Ingenuity or IPA, but I prefer the Metacore platform. They do very similar things, but these platforms are only as good as the database that they're supported on. And for hematology research, I tend to get more consistent answers using Metacore. So when we did an enrichment analysis for those 200 odd genes, what we found at the top of this list, or near the very top, is number 2. You can't quite read it, but effectively it's saying the development of GCSF-induced myelodifferentiation is one of those pathways that are significantly enriched in your data set. And this sort of goes back to confirming our earlier data that indeed, if you overexpress Runx-Bonito, you've got this dysplastic granulopoiesis pathway. And when you look at pathways, because pathways and enrichment analysis are 2 different things, it comes up with a very similar answer. So we were really pleased with that. It sort of showed the approach that looking at the proteins was consistent with looking at mRNA expression. Now, unlike our mRNA data, where we sort of did an unsupervised analysis and looked for changes throughout the whole genome, we really particularly focused on transcription factors here. And that's because we thought if it's a block in differentiation, this might be mediated by transcription factors or regulators of transcription. So we performed a transcription factor overconnectivity analysis. So this looks within your data set to see which transcription factors might be regulating other genes. And we combined that with our mRNA data as well. And what we identified were 4 proteins. And this was also good because we're identifying CBF beta, which we know is really important in RUNX1 leukemias, and PU1. And these are transcription factors that have previously been shown to be disturbed in 821 disease. But what was also pulled up with this was this CEBP beta. And this was repressed upon expression of Runx-Bonito. And this is something that hasn't really been reported before. So Alexa has been following up on this, and hopefully she'll put this into publication shortly. So to show you some of that data quickly for the last 5 minutes, what is CEBP beta? Besides being really hard to say all the time, it's a CCAAT enhancer binding protein. It's a family comprised of 6 members, ranging from alpha to epsilon. and they promote the expression of certain genes through interactions with their promoters. So what you get is involvement in lots of different processes, particularly differentiation and apoptosis. And that's because they can recruit co-activators such as CBP, and in turn they can open up that chromatin structure and recruit more basal transcription factors. So we're really interested in this, not only for its own role, but there is a lot known about CEBP alpha in AML, and that I think indeed there are mutations in alpha that one of the sort of stratification mutations in AML. So it's interesting that beta hasn't really been looked at. So when you look at beta, I'm just going to call it beta, it's easier in terms of its mRNA expression, and we use online data sets for this. You can see that it's expressed at the hemopoietic stem cell level, but as cells differentiate, so as you go from left to right, they're becoming more monocytic or granulocytic, they upregulate the expression of this gene or protein. So the first thing we did, we took our CD34 cells, or Alexa did, and she overexpressed beta in that system. And what she's found was that there was an increase in granular poiesis, probably at the expense of making monocytes. But the problem is the fact that overexpression of beta promoted differentiation suggests that its upregulation is not really likely to promote leukemogenesis. So we're sort of a little bit disappointed in this result, I guess, because it's not really that interesting. So we were much more interested in what would happen when you knock down expression in CD34 cells. And I haven't shown you all the validation of that, but effectively black is our control, and the other 3 lines are mission shRNA, of which we had about a 50% knockdown. And whilst we had a reduction in growth, we didn't really see any changes in the phenotype of those cells in terms of differentiation. And in fact, if you look at the morphology, they seem to be quite normal. I think this is sort of preliminary data, if you'd like. This latter observation could probably be explained by the lack of complete knockdown of beta. That's one explanation. Or it could be that given that this is a family of 6 different proteins, maybe there's redundancy in the context of the other CEBP family members. So to summarize that aspect, I think what you can say is we've identified 256 proteins dysregulated by RUNX1-ETO, and of those, beta is something that's important. It might be upregulated during granulocytic development, and at the moment we can say knockdown had little effect. But I haven't had time to show you that data, but it seems to be important in the context of transformed cells. So when you look at RUNX1-ETO in cell lines, there's definitely, if you knock down CEB beta in transformed cell lines that don't express RUNX1-ETO, then we do get changes in phenotype. And that's something that Alexa would currently explore if she was staying here longer. So we need to do a bit more work on this, I think, to get that to the level of publication. So I think I'm on 50 minutes time. So I just need to particularly thank Richard, Steve, for their continued collaboration in all our work, Alexa for the mass spectrometry data, and the other guys in blue contributed to the CD200 study. And Professor Wetton in Manchester and Andrew Pierce, they're our experts for mass spectrometry. So I know that was a bit of a whistle-stop tour. I hope it was informative and broad enough for everyone to understand, and happy to take some questions.
Thanks, Alex. That was a fantastic overview of AML and two decades plus of research interest and activity. Thanks particularly for showing a picture of me when I had slightly more hair than I do at the moment. That was very nice of you.
Last year, Steve.
So please feel free, everybody, to contribute questions through the chat. I'm at 10 to 4, so we've got about 10 minutes potentially for questions to Alex. So as is kind of traditional, while you're thinking of questions, I'm going to ask one just to sort of kick things off. So, I mean, obviously, RUNX1 has lots of different partner translocations and different ways it can be deranged in AML. And I mean, it's really interesting that they seem to have so many different prognostic connotations. So you've talked a lot about RUNX1-ETO, which is generally considered a favorable risk abnormality. RUNX1 mutations are generally fitted in the adverse risk category now. Do you have any kind of thoughts about how those different, how it's deranged in those different mutations and translocations, which sort of lead to it having those different connotations in how the disease works out?
Yeah, so if you look at the biology of where that translocation happens in terms of what components of RUNX1 remain or what components come in from the other translocated product, then you have vastly different responses. So I think in at least ALL, for example, most things are happening to the left of the N-terminus of RUNX1. So you have a number of different domains that appear there that are not relevant to the myeloid component. And what you've got in RUNX1 ETO, you've got 4 domains. They're called the nervy homology domains. And each one of those domains respond differently to recruiting different cofactors. So in all those abnormalities that you talk about, you're losing these domains or gaining domains. And each one of those recruit different transcription factors and cofactors, which ultimately will lead to a change in the phenotype. I don't know if you're aware, there's a particular abnormality called RUNX1 9A. And that's important because basically when you put RUNX1 full length into a mice model, they don't really generate leukemia. So it's quite interesting in that you've got this molecular abnormality that we say it's leukemia generating, but in mice, you really need to give them sort of more ENU or more mutagen to get that overt leukemia. But when you put 9A in, this lacks a lot of those nervy homology domains. Those mice have a rapid leukemia. So, I think that just exemplifies how those domains, those nervy homology nervy domains are really important in the translocated product.
Thanks, Alex. I can see there's a question in the chat from Awen. Have you tried to reinterrogate your transcriptomic data using the proteomic data and vice versa? Might this let you refine and get the best out of both data sets?
Yes, it is the answer to that's what we've tried to do in the past in general. And with IPA and Metacore, there are a couple of algorithms that allow you to identify the upstream regulators, if you like. So if you put in your mRNA gene targets and put in your protein targets, there are algorithms now that can combine that data to make it a little bit, more comprehensive. We didn't really get much success in that approach, to be honest, and it was quite tricky to try to explain in the paper. So in the end, we just went with, this is the highest fold change in gene of interest, and it was a bit easier to sell the story.
Thanks, Alex. We've got another question. Not sure whether this is from Owen as well. Anyone looked at the epigenetic effects of RUNX1 ETO?
Yeah, so as you know, AML is really a disease of epigenetics, isn't it, I guess? And there's a lot of rationale to use epigenetic drugs in patients with AML. Now, the co-repressor complex recruits histone deacetylases, which are sort of epigenetic themselves. So there's been a lot of work going on about RUNX1 ETO and inhibitors such as, I think, Verinostat, if I've said that right, Steve, and how they might treat patients with RUNX1 ETO. They've been variable successes. I don't think much has gone on with that, given that those studies were around 2005 or 2008, something like that. So that's 15 years ago, and nothing's progressed with those HDAC studies. But there's a lot about epigenetics and the chromatin accessibility for RUNX1 ETO. And if you are really interested, check out, I forgot her name now, a group in Birmingham. This is their type of work that they do quite a lot. I forgot her name. It'll come to me in a minute.
Yeah, I think we were involved in a study with Verinostat in myeloproliferative neoplasms. I think one of the problems was, and it's a kind of bit of a class effect with the HDAC inhibitors, was there's a lot of gastrointestinal toxicity, and it's been very difficult to sort of find good therapeutic windows for that class of drugs, I think. I mean, on that subject, you mentioned CEBP beta. In terms of kind of targetable abnormalities and potential inhibitors for sort of as offshoots from this work, where do you see the most sort of cancer targetable
Yeah, I think that's a really good question, Steve, I have to say. And I don't know if I've got the right answer for that. I think the problem is those ones are quite key regulators. And I think if you're going to mess with this, you're going to mess with quite a lot of different stuff. So I think you really got to focus down to the pathway that beta is really regulating. And I think people have tried to look at alpha and targeting that, and that hasn't been so straightforward either. So I think we need a bit more work to understand what beta is really doing. And like I said, if basically in 821, it's repression. So we can't really target a repression. We would need to target something that it upregulates, I guess, to make that more applicable in at least 821. What you wouldn't want to do is inhibit something that seems to be important for normal hematopoiesis as well. Although saying that, our knockdown studies had no effect. So, I don't know.
Thanks, Alex. So that's excellent. I'm not seeing any more questions in the chat box or the Q&A at the moment, and we are sort of ticking around towards 4 o'clock. So that's probably a good time to sort of start wrapping up. So thanks again for a brilliant talk. Thanks to all of you for dialing in, because I know this time can be tricky on a Friday afternoon. So it's great we've had such a good audience today. And on that subject, just to sort of get some advanced publicity in for the next one of these, which is taking place in 5 weeks' time. This was originally going to be next week, but we moved it forward a week because of being aware of October half term next week. So the next one of these webinars is going to be on Friday, the 26th of November, same time. And this time we're going to be welcoming Professor Aled Clayton, also from Division of Cancer and Genetics, who's going to be speaking about